Hi, my name is Tom OsbornPopp, and I'm an Arnold O. Beckman postdoctoral fellow in chemical instrumentation at Rutgers University. As of September 2023, I will be an assistant professor in the Department of Chemistry at Oregon State University. In this video, I will be sharing with you the results of my postdoctoral research at Rutgers, where I developed new solid-state nuclear magnetic resonance technologies to study the biochemistry of deep-sea microorganisms.
As someone who is passionate about developing new instrumentation to study chemical phenomena, I'm honored to be supported by the Arnold and Mabel Beckman Foundation to pursue my research goals. I'm constantly inspired by Dr. Arnold Beckman, whose pioneering and impactful contributions to scientific instrumentation include the pH meter, the spectrophotometer, the oxygen analyzer, and many others. The ethos of his work was centered around the betterment of humankind, both through the technology that he developed, but also through his enduring impact on future generations of scientists like myself, through the generosity of the Arnold and Mabel Beckman Foundation. In my career, I aim to follow in his footsteps to develop new tools that can answer the challenging research questions of our modern era, and in addition, I aim to educate and support other young scientists in this effort as well. Now I'd like to get started telling you about the research I did developing new instrumentation for solid-state NMR studies of thermopiezophilic bacteria. Thermopiezophiles are microorganisms that grow optimally under high temperature and pressure conditions. This means that they're actually quite difficult to study in the laboratory, which means that we need new tools and techniques to understand them. The organism that I studied as part of this project is called Nautilia abyssei strain PV1. It's a chemosynthetic thermopiezophile that can grow at both low and at high pressures. Chemosynthesis is a process that can be thought of as photosynthesis without light, meaning that these organisms use CO2 as their carbon source, but instead of using sunlight to power this process, they use inorganic molecules such as sulfur or nitrate. PV1 was originally discovered in 2014 during an expedition to the East Pacific Rise 9 North, which is a hydrothermal vent feature off the coast of Mexico. On this expedition, the remote-operated vehicle Jason was used to collect samples at a depth of 2,500 meters below the surface of the ocean, which corresponds to 250 bar of hydrostatic pressure. PV1 was isolated from hydrothermal vent fluids associated with the polychaete worm Alvinella pompeiana, which has filament-like hairs to support growth of microbial biofilms, and it may be a site where PV1 grows naturally. After isolation of PV1 into a pure culture by members of Professor Costantino Vetriani's lab at Rutgers, I was able to grow it as a batch culture at a temperature of 55 C and at 2 bar pressure, which is a relatively low pressure compared to the pressures it had experienced in its natural habitat. We also grew PV1 at pressures exceeding 200 bar, using a high-pressure chemostat. The fact that PV1 can grow under both low and high pressure conditions makes it an ideal case to investigate what biochemical changes its phospholipid membranes must undergo in order to adapt to these extremely high pressures. In response to an external stimulus, such as a change in temperature or pressure, bacteria will modify the composition of their lipid membranes in order to maintain a certain level of membrane fluidity, which is known as the homeoviscous response. Many biological processes require fluid membranes in order to function properly. To use an analogy, an organism that is adapted to room temperature might have membranes with consistency of olive oil. If that organism is subjected to higher temperatures, their membranes may become too fluid to function properly. Therefore, in response to the increase in temperature, it will increase the number of saturated tails in its phospholipids in its membrane in order to be more like that of coconut oil, which is solid at room temperature, but fluid at higher temperatures. Phospholipids are molecules with 2 hydrocarbon tails and a polar head group. This one, shown here, has a 16 carbon tail and an 18 carbon tail, and that 18 carbon tail has 1 degree of unsaturation. The head group is known as phosphatidylethanolamine, or PE, but other head groups can also be found in bacterial membranes, such as phosphatidylglycerol, or PG. The homeoviscous response is relatively well understood for a temperature stimulus, but is less well understood for a high-pressure, high-temperature stimulus. Therefore, my aim in this project was to understand how lipid composition in thermopiezophiles is related to membrane fluidity under high temperatures and pressures. The way I did this was by first culturing PV1 in the lab under both low and high pressure conditions. then quantifying which phospholipid species were present in their membranes using mass spectrometry-based lipidomics. To better understand these complicated lipid profiles, I then synthesized phospholipid vesicles, or liposomes, with simplified compositions. I examined these model samples using high-pressure solid-state NMR to measure the mobility of these lipids under low and high-pressure conditions. But first, what is NMR? And how can we use it to study membranes at high pressure? Nuclear magnetic resonance, also known as NMR, is a technique that provides element and chemical site-specific information about samples of interest by directly probing transitions between nuclear spin energy levels. We take our sample and then put it into an NMR probe, which is then housed in a superconducting high-field magnet. Common nuclei of interest include hydrogen, which is also known as proton, carbon, nitrogen, and phosphorus. When I studied my lipid samples, I measured proton and phosphorus NMR spectra. In order to study solid or solid-like samples such as phospholipid membranes with NMR, we need to perform a technique known as magic angle spinning, where the sample is placed into a rotor which spins pneumatically at an axis inclined at the magic angle, or 54.74 degrees, with respect to the magnetic field. This technique averages away spin interactions that broaden NMR spectra, yielding highly resolved spectral lines that allow us to observe each of the phospholipid species present in our sample. To perform high-pressure magic angle spinning experiments, I used specialized high-pressure sample rotors, which enabled me to pressurize samples up to 300 bar or more using gas as the pressure transducing medium. This pressure is maintained while the rotor spins within the magnet, allowing us to observe by NMR the properties of the sample under these high-pressure conditions. To actually pressurize these rotors, I developed a high-pressure system that allows for the pressurization of a high-pressure vessel using a manual high-pressure pump. This can actually be a bit of a workout, but it significantly reduces the cost and complexity required to reach such high pressures, making high-pressure experiments very accessible. I use this apparatus to pressurize sample rotors with helium gas up to 250 bar. Let's now take a look at the phospholipid profiles of PV1's membranes when grown at low pressure and when grown at high pressure. At a glance, you can see that the lipids can be categorized by which head group they have, with the membranes being primarily made of PE, with nearly 10% PG present. These categories can be further broken down by tail group composition, with a higher diversity of tails in the higher pressure lipid profile. Rather than making model membranes composed of many lipids with an arbitrary variety of lipid tails, I sought to first understand what role the head groups can play in membrane fluidity, and why the head group concentrations don't appear to change much between the two conditions. I synthesized two liposome samples containing differing ratios of PE and PG, one with a 75 to 25 ratio of PE to PG, and another with an 87.5 to 12.5 ratio of PE to PG. This is a cryo-EM image of one of the samples, showing that they are multilamellar vesicles with a range of diameters. I examined both of these samples at 1 bar of pressure and at 250 bar of pressure. First, by taking proton solid-state NMR as a function of temperature to measure the gel-to-fluid phase transition under each condition. Below the phase transition, the proton signal arising from the lipid tails is very broad, but above the phase transition, the peaks narrow and increase in intensity. By plotting the derivative of intensity with respect to temperature, I can measure the phase transition temperature. The key takeaway here is that 250 bar of pressure increases the gel-to-fluid phase transition by 5 degrees Celsius, regardless of the lipid composition. We can also see that the addition of PG acts to reduce the phase transition temperature regardless of the pressure condition. Doubling the amount of PG in the membrane decreases the phase transition temperature by 4 degrees Celsius, both at 1 bar and at 250 bar. I also examined the fluidity of these membranes at 55 C, the optimal growth temperature of PV1, by performing a special solid-state NMR experiment known as center band-only detection of exchange, or CODEX. The CODEX experiment generates a signal decay curve from which the lateral diffusion coefficient for each phospholipid can be extracted. On the left, you can see the signal decay curve for PE in the 12.5% PG sample. And on the right, I've plotted the lateral diffusion coefficient for each species under each condition extracted from these decay curves. What we can take away from these results is that high pressure reduces the lateral mobility of all phospholipids, even in a fluid phase membrane at 55C, and that PG can increase membrane fluidity even under high pressure conditions. So now let's go back to our lipidomics results. If PG is such a potent membrane fluidizer, why doesn't PV1 change its concentration of PG under high pressure conditions? PG is a negatively charged head group, and changing the amount of PG in the membrane will also change the charge balance within it, which could potentially alter critical biological processes. So now we return to the increase in diversity of lipids that we see at high pressure. It turns out that this actually represents an increase in shorter length lipid tails. The average lipid tail length for the membrane goes from 16.96 at low pressure down to 16.22 at high pressure. Shorter tails are more mobile than long ones under any conditions, and so we propose that the mechanism by which PV1 maintains membrane fluidity under high pressure conditions is by reducing its average lipid tail length. And with that, I would like to thank both of my research advisors, Professor Newcoop and Professor Vetriani, as well as the rest of my colleagues at Rutgers for their support. Finally, I would like to sincerely thank the Arnold and Mabel Beckman Foundation for providing me with the opportunity to pursue this interdisciplinary research. It would not have been possible without their support. Thank you.